Welcome to the Scientific Dose. Today, we're embarking on a real biological thriller. We're going to trace the intricate cause and effect journey of our cardiovascular health. We're looking at atherosclerosis here, and not just as some general hardening of the arteries that you might have heard about. No, we're looking at it as a dynamic, microscopic battleground where our body's own defense mechanisms kind of inadvertently turn against us. It's a fascinating story of lipids, cells, and a biochemical cascade that can, quite literally, dictate human life. Here's our roadmap for this journey. We'll start with lipoproteins and lipid metabolism, move on to healthy versus damaged endothelium, look at macrophages and foam cells, map out the timeline of plaque formation, and finally wrap up with plaque rupture and thrombosis. All right, let's get moving. Section 1, Lipid Proteins and Lipid Metabolism. Okay, let's dive into this and introduce the core biochemical characters of our story, the lipids and the vehicles that carry them. Now, because lipids are hydrophobic, meaning they repel water, they can't just dissolve in our watery bloodstream. They need a ride, right? And those transport vehicles are our lipoproteins. Think of it as a biological tug-of-war happening right inside your veins. On one side, we have LDL, or low-density lipoprotein. Its main gig is delivering cholesterol from the liver to your peripheral tissues. But when there's way too much of it, it starts dumping this cargo into the arterial walls, which is exactly how it earned its reputation as the bad cholesterol. On the flip side stands HDL, or high-density lipoprotein. HDL is like the biological cleanup crew. It performs something called reverse cholesterol transport. Basically, it actively scoops up excess cholesterol from your tissues and hauls it right back to the liver. And that's why we proudly call HDL the good cholesterol. Now, what's really interesting about this slide is the actual molecular anatomy of a lipoprotein particle and how it's perfectly constructed to solve that solubility problem we just talked about. Right in the middle, you see the hydrophobic core, those tightly packed yellow and pink spheres. That's where the precious cargo of triglycerides and cholesterol esters are safely tucked away from the water in the blood. Then, surrounding that core is a hydrophilic, or water-loving, outer layer made of blue phospholipids and free cholesterol. And wrapping around the whole surface, those are the large green structures known as apolipoproteins. These are absolutely essential, guys, because they act like shipping labels and functional keys for the entire particle. To truly grasp how this entire system hums along, we've got to understand that every single component here acts as a highly specific manager regulating lipid metabolism. Let's break it down. First, ApoB100 serves as the main structural protein of LDL. It's the specific ligand that gets recognized by LDL receptors so your tissues can actually take up the cholesterol. Meanwhile, ApoC2 acts as a powerful activator. It literally turns on an enzyme called lipoprotein lipase, or LPL. Then we have ApoE, which functions as a sort of homing beacon to mediate the liver's clearance of leftover lipoprotein remnants floating in the blood. And speaking of those enzymes, once LPL is activated, it goes to work aggressively hydrolyzing triglycerides into free fatty acids and glycerol to give your tissues energy. Finally, we have LCAT. This is an enzyme that's totally crucial for HDL maturation. It actively converts free cholesterol into cholesterol esters so they can be packed tightly into the HDL core for a safe trip back to the liver. Honestly, it's just a perfectly calibrated machine. Section 2, Healthy versus Damaged Endothelium. Let's move to and see how this builds into the physical setting of our blood vessels and witness the true inciting incident of our biological conflict. Take a look at the left side here. In a normal, healthy endothelium, which is the inner lining of the artery, vascular permeability is so tightly controlled. The cells form a really strong barrier. They naturally promote vasodilation to keep the vessels nice and open, all while actively maintaining an anti-thrombosis and anti-inflammatory state. It's a peaceful, highly functional environment. But look over to the right. When exposed to persistent injury, maybe from the sheer physical stress of hypertension or the toxic load of high cholesterol, that barrier just breaks down. The endothelium gets damaged and dysfunctional. This leads to sudden vasoconstriction and increased vascular permeability. So instead of actively repelling clots and immune cells like it's supposed to, this damaged endothelium actively promotes thrombosis and intense inflammation. Section 3, Macrophages and foam cells. With our arterial barrier compromised, the environment is now totally primed for a full-scale invasion. Here's the twist. The true villain in this story isn't actually just regular LDL. It's oxidized LDL. When too much LDL enters that newly permeable, damaged intima layer, it gets stuck. It gets trapped. Down there, it's exposed to reactive oxygen species generated by nearby stressed-out cells. This oxidative modification physically alters the normal LDL particle, so it's no longer recognized by normal cellular receptors. Instead, it becomes highly atherogenic and intensely pro-inflammatory. It effectively rings a massive biological alarm bell, marking the transition to inflammatory activation and forcing the damaged endothelium to release pro-inflammatory cytokines like IL-1 and TNF-alpha. That alarm bell triggers a cascade. First, monocyte recruitment kicks off via adhesion molecules. These white blood cells literally squeeze their way through the compromised barrier, migrating directly into the intimal layer where they differentiate into active macrophages, basically gearing up to clean up the mess. But as they attempt to engulf the dangerous oxidized LDL, they use specific scavenger receptors on their surface, mainly SRA and CD36. So the crucial point is that unlike normal cholesterol receptors, these scavenger receptors are completely unregulated. The macrophage literally has no off switch. It's just going to endlessly engulf the oxidized LDL until it becomes absolutely engorged with lipid droplets, transforming into a toxic, bloated foam cell. And these foam cells, they are the primary driver of plaque growth. Section 4. Timeline of plaque formation. Now that we've met the cellular players and seen exactly how they malfunction, let's map out the chronological progression of this disease over time. This progression of atherosclerosis really is a step-by-step biological sequence. It always begins with that initial endothelial injury which serves as the trigger. This pretty quickly leads to the development of a fatty streak, as those accumulating foam cells form the earliest visible lesion right there in the artery wall. Then, as dying foam cells release growth factors, smooth muscle cells are prompted to migrate into the intima and intensely proliferate. Finally, in a desperate attempt to contain the damage, these smooth muscle cells synthesize extracellular matrix proteins, like collagen, to build a thick, stabilizing fibrous cap right over the toxic lipid core. Just watch how this visual chronological progression unfolds inside the artery, moving from left to right. It starts out as a completely normal vessel with a nice, wide-open lumen, but soon a subtle accumulation of yellow marks the fatty streak forming just under the inner lining. As smooth muscle cells migrate and foam cells continue to die off, a fibrofatty plaque physically expands. Eventually, an advanced plaque develops with a thick fibrous cap, but notice how the actual lumen, the critical space for blood to flow, becomes severely, dangerously narrowed. And ultimately, this timeline leads to the catastrophic endgame. The plaque ruptures inside the vessel. A massive red thrombus forms instantly, and the artery is completely occluded. Section 5. Plaque rupture and thrombosis. This brings us to the absolute climax of our biological thriller, the terrifying transition from a slow-growing plaque to an immediate, life-threatening state. To really understand the stakes here, just contrast that unimpeded, healthy blood flow with an artery that is heavily narrowed by an advanced atherosclerotic plaque. You can clearly see the yellow core trapped right beneath the fibrous cap. That core, it's a highly toxic, necrotic graveyard. It's absolutely packed with dead foam cells, aggressive macrophages, cellular debris, and rigid calcium deposits. The artery is literally being choked. But as incredibly dangerous as the narrowing is, the real threat here is the stability of that fibrous cap holding all that toxic core back from the bloodstream. So how does a stable plaque become a lethal event? It all comes down to the mechanisms of plaque instability. Inside that core, continuous chronic inflammation means immune cells are constantly releasing enzymes that actively chew up and degrade the stabilizing collagen. This causes severe thinning and weakening of the fibrous cap. At the exact same time, foam cells keep dying off, which expands the necrotic core of lipids and dead cells, building up tremendous internal pressure. It's a ticking time bomb, guys. The cap gets thinner, the core gets bigger and bigger, until the cap finally snaps, abruptly exposing that highly thrombogenic subendothelium material directly to the flowing blood. And this brilliantly illustrates exactly what happens when that physical barrier breaks. The highly reactive subendothelium matrix, specifically collagen and tissue factor, is suddenly exposed to the bloodstream. The body basically panics, thinking it's bleeding to death. Platelets immediately aggregate to the exposed surface, the coagulation cascade violently activates, and a massive red thrombus forms instantly. This clot can grow in literally seconds to minutes, severely impeding or even completely stopping blood flow to the heart or brain, which is exactly what results in a heart attack or stroke. We've traced the whole journey, from a single oxidized lipid all the way to a catastrophic arterial blockade. It's an incredible, yet frankly terrifying cascade of biological cause and effect. But it leaves us with an absolutely critical thought to ponder. Given the fragile biochemical balance that dictates the stability of these plaques, how profoundly do our daily lifestyle choices, our diet, our exercise, our stress levels, alongside our genetics, dictate the ticking clock of atherosclerosis within our own bodies. The science shows us the exact mechanism, but the timeline, well, that is often in our hands. Thank you for joining me on this microscopic journey.